Today we're gonna be doing lecture 2, which is blood and lymphatic system. I just turned on live transcript, so if you need the caption, you can turn that on your computer. All right, so let's get started. First question here: list 3 types of granulocytes. So let's explain what granulocytes are. These are white cells, WBCs, with visible granules. Okay, and granules are kind of these similar to lysosome, if you would. They're little vacuoles that has various chemicals in them. Okay, and you can see them clearly for granulocytes. So white blood cells with visible granules under the microscope. Okay. Oh, by the way, like if for whatever reason Zoom kick us all out because the email said they're experiencing intermittent issues and we cannot come back, then just check the email. Okay, like I'm gonna send out like a Google link. If we cannot join Zoom or something, then I'm gonna send out a Google link and we can go meet on Google instead. Okay, just in case it happens. So 3 types of granulocytes. Does anybody remember some examples of granulocytes? Help me out here, please. Yeah, Brian said neutrophil. Someone else says basophil. Excellent. So neutrophil, basophil, and Donna said eosinophil. That's correct. Okay, so neutrophils, they are a type of phagocytes. Right? Phagocytes are cells that can go around and do phagocytosis. They can eat up other cells. And neutrophils is actually the most abundant type of white blood cells. You have the most most of your white blood cells are neutrophils. They account for about 70% of all your white cells. So they are the ones that will go around and eat up things that are not supposed to be in the body. Basophils, they are responsible for creating the inflammatory response. Okay. And eosinophil, they are important for fighting parasite. Okay, so these are just very basic functions. Of course, they do much more than this, but that's all we have to know for now. Okay, so the opposite of granulocytes are agranulocytes. Now, they also have granules, but you just can't see them under the microscope. Okay, they also have granules, but they are not visible under the microscope. Okay, they don't take up the stain as well as the granulocytes. Okay, but doesn't mean they don't have it. 3 types of agranulocytes. Anyone? 3 types of agranulocytes. Okay, thank you. There's monocyte, which can transform into macrophage, right? The big eater. So monocyte just circulates and around the body and do surveillance. And when they encounter something foreign, something that's not supposed to be there, they can differentiate, they can transform into macrophage. Okay. People mention T cells and B cells. Absolutely. T cells are responsible for coordinating immune response. B cells are responsible for antibody production. Okay, let's match the cells with the function. Okay, responsible for inflammatory response, and that would be the basophil. Okay, now I just want to mention that basophil has a relative, okay, that you will see later on. I just want to mention it now, so it wouldn't be a surprise when you see it. This is related to something called mast cell. Okay, so both mast cell and basophils will play a role in inflammation. Okay, the basophil circulates around the body. Mast cell, they are stuck to tissues. Okay, so when you cut your finger and it start to become inflamed, right? It's red, it's swollen, it hurts, and it feels warm. All those things are characteristics of inflammation, and that's because of the mast cell releasing histamine. Okay, so basophil also will release histamine, but they're more for the entire body reaction, okay, like systemic reaction. Okay, just keep that in mind. Coordinate immune response, that would be the T cells. Okay, there are different types of T cells that you will learn next lesson. Can differentiate into phagocytic macrophage. We mentioned that earlier, right? These are the monocytes. Okay, sometimes in places of the body where there's a lot of foreign antigens coming in, foreign cells coming in, you would have what we call resident macrophage just staying there, right? Because it's so prone to getting infection, and such as the lungs, right? You breathe in all these things all the time that are from the outside, and you want to have some macrophage staying at the lung tissue at all times. Okay, but sometimes, right, in other places that are not as vulnerable, then the monocyte would visit them, and only if they encounter something foreign would they transform into the macrophage. Next, attack parasites. That would be the eosinophil. Like some things are too big to phagocytize, to engulf, so you need to secrete chemicals to attack them. That would be the job of eosinophil. Most abundant phagocytes, that would be the neutrophil, and finally produce antibodies. Okay, that would be D right here, antibodies, the B cells. All right. So I just want to clarify antigens versus antibodies, right? So this is a cell. It could be any cell. It could be your cells, could be an outside cell, could be a virus, could be bacteria. And all cells, they have antigens on them. Okay. So antigens are just molecules that are found on the surface of a cell. They could be proteins, they could be carbohydrates, lipids, combinations of those things. Okay, so these are antigen. Okay, antigens are not a bad thing. They are just part of a cell. Now, what happens is your body knows about all your antigens. They will not attack them. But if something from the outside goes into your body and they have different antigens, then your immune system will react and it will attack those foreign antigens. Okay, and the way they attack it is they will make antibodies that would tag these antigens. Okay, so that's antibody. All right. So just to recap, all living cells, all cells, we say all cells have antigens on them. Okay. You have antigens, I have antigens, bacteria have antigens. When you have foreign antigens, okay, cells that contains antigens that you do not have, right? We call them foreign antigens. Then the antibodies will bind to them and it will attack them. Are we all clear on the difference between antibodies and antigens? Okay, thank you for the confirmation. All right, next we going to move on to this concept of coagulation. When you have a cut, a damaged blood vessel, how does the bleeding stop, right? So when the vessel is damaged, the underlying tissue is going to release something called thromboplastin. Okay? When you do not have a cut, this stuff will not be in your blood. Okay, this will only goes into your blood when you damage a vessel and the underlying tissue is exposed. So the thromboplastin will convert prothrombin. Okay, prothrombin is inactive and it's part of your plasma protein. It just circulates in your blood. But in the presence of thromboplastin and a little bit of calcium, prothrombin will get activated into thrombin. Okay, this is the active form. The active form. Okay. Now, in your blood, in the plasma, right? In addition to prothrombin, you also have something called fibrinogen. Fibrinogen, which is also inactive. But in the presence of thrombin and calcium, fibrinogen will get converted into fibrin. Okay, so that's active. And what the fibrin does is they will form these long strands, okay, which will kind of form a net. And the net will catch things like platelets, right? It will catch other red cells. And eventually it will form a plug that seals the break in the blood vessel, and it will stop the bleeding. Okay, so this is a fibrin clot. Okay, so one thing leads to the next, right? It's kind of like dominoes. So again, when you are not injured, when you don't have a cut, you will have prothrombin and fibrinogen circulating in your bloodstream, but they're not active. Okay. Once you have a damaged vessel, thromboplastin will come out and it will initiate this process. Okay. So this is one of the several coagulation pathways that exist in the body. Moving on. Complete the following chart related to ABO and RH blood group. Can someone explain to me what is the meaning of blood groups? Anybody can help me out here? Feel free to unmute or type in.
Antigens and antibodies.
Part of what you said is correct. Okay, so thank you, first of all. Blood groups, okay, they are the presence or absence of antigens on the surface of red blood cells. Okay, and Hammer said group type AB. Yeah, so A and B is just one of the blood group, right? There are many different blood groups. I think they found like 42 or something now. Okay, and the most common one that we look at when we do blood transfusions would be the ABO blood group, the RH blood group. But like I said, there are many other, right? MN, something called Duffy, something called Cal. There are so many different blood groups. It's just that these are the major ones that people are most familiar with. Okay, so again, the blood group is whether or not you have certain things on your red cells. And so if you have A on the red cells, then you have type A blood. Right? If you have A and B, then you have AB blood. Okay, so this has nothing to do with antibodies. It's just talking about what type of antigens, what type of molecules you have on the surface of the cell, of the red cell specifically. Okay, so alleles are different versions of a gene. So for example, eye color is a trait, and you can have the brown eye allele or blue eye allele, right? Different versions of the gene that give rise to different versions of the trait. So similarly, for the blood group, we're going to have different alleles for it. We're going to have different versions of the ABO blood group. So the three alleles that we talked about in class included IA, IB, and small I. Okay, so there are a total of 3 alleles for ABO. Okay. How many possible genotypes do we have for the ABO? Does anybody remember? 6. That's right. So we are diploid. We only have 2 sets of chromosomes. So even though there are 3 alleles in the population, at most we can have 2 alleles, right? So they could be IA, IA small I, IB, IB small I, right? Little I, and IA, IB, right? So a total of 6 possible genotypes. Okay. Again, all these are explained in a lot of details in the recordings. So if you haven't had a chance to watch them, please do so. And phenotypes would be the expression of the genotype, right? So in other words, if you inherited the IAIA gene, what actually would be your blood type? So both IAIA and IA small I would be type A blood. IB small I is type B blood. Little I, type O. IAIB is AB. So with the 6 genotypes, we can have total of 4 phenotypes. Okay, and the reason for that is because IA and IB. How would you describe the relationship between these 2 genes, IA and IB? What's the best word we can use to describe them? Think back to like genetics unit in last semester. If you have IA and IB together, how do they affect each other? Is one dominant over the other? You know, what's the word I'm looking for? Okay, that's okay. Yeah, Donna said they're both dominant, right? So they are co-dominant, in other words, right? Both of them will show up. Okay, so IA, IB are dominant over small I. That means they will cover up small I, right? And IA, IB are co-dominant with each other. Okay, so when you have both will show up. When you have them in combination with small I, they will cover it up. So that's why IA small I is still type A blood. Okay, whereas little I will give you type O, right? Little I is the absence of A and absence of B, and as a result, we call you type O. Okay. What about RH? How many alleles are there for RH? Anyone? Yep, Tina, Donna, both are correct. We have the positive allele and the negative allele. Positive allele and the negative allele. Right? So if you're positive, then you can make the RH protein and put it on your red cells. If you're negative, then you cannot make it. Number of possible genotypes. Positive, you can have 2 copies of the RH gene. You can have 1 copy or you can have none. Right? So positive will give you RH positive blood type. Positive negative will also give you the same thing. Whereas the negative will give you RH negative. Okay, so if you're RH negative, that means you do not have the RH protein on the surface of your red cells. Are we together? Looking at the chat to see if anyone wants to ask anything. Okay. Great. Now I know I did this chart already, but probably it's a good idea to go through it again. Okay, if you've done it already, we'll just review it together. All right. Complete the following chart related to ABO blood group. All right, so this is only ABO blood group. We are not talking about RH for the moment. Right, and the key thing here to know is this. We'll write down. Let's write down in pink right here. Right, your immune system will create antibodies to attack any foreign antigen. I mentioned this earlier, right? But let's just put it in writing. Okay. Any foreign antigens. So if your body detects an antigen that you do not have, you will make an antibody and you will attack it. All right. So next week, you will learn about vaccination, right? So when you inject the vaccine into your body, right, you introduce a foreign antigen, and in turns, your body will create antibodies. So when the real virus or real bacteria comes, you already have the antibodies in your system, and you are able to fight it off much more effectively. Okay. So let's just quickly recap where the antigens and antibodies are. When you spin down the blood, you collect blood from someone, and then you put it in a centrifuge. Okay. What do we call this stuff at the bottom? What do we call that? Yeah, that's where the RBCs are, right? Okay, the hematocrit, right? We call it the hematocrit. Okay, and that makes up approximately like 45% of your total blood volume, right? What about the stuff here at the top, the yellowish clear stuff? Yep, that would be the plasma. Plasma. So where do we find the antibodies? Do we find it in the plasma, or do we find it in the hematocrit? Where do we find the antibodies? Okay. All right, let's do a vote. Okay. Let's see what people think. Launch this. Okay. If you think the antibodies are in the plasma, please choose A. If you think it's in the hematocrit, please choose B. I'm asking you, where do you find the antibodies? Okay. Very good. Just give me one second. By the way, do we have a Samira here? Samira Farah? Are you here in the session right now? Okay. Never mind then. If you are, please send me a private message. I need to talk to you about something. All right, this is the result. 10 of you say it's in the plasma, 8 of you say it's in the hematocrit. The antibodies are found in the plasma. Okay, these are the antibodies. Antibodies are Y shape. Okay. The hematocrit is where you will find the red cells, and the red cells, the surface of the red cell is where you will find the antigens. Okay. So antibodies found on plasma, found in plasma, is floating around plasma. And then the antigens are on cells, right? And when we talk about like what we're talking about in this particular context, the antigens are on the surface of the red cell. Okay, even though all cells have antigens, right? I have to emphasize that. Your liver cells, they're gonna have antigens. That's why we cannot just take someone's liver and then transplant it into the other person, right? Because the antigens on the liver cell will be rejected. It's not the same. Okay. Does anybody in this world have the exact same antigen? Will two people ever have the same antigen? No? Anyone else want to chime in on that? The only person who would have the exact same antigen as you is, yeah, exactly. If you have an identical twin, okay? Identical twin, they share everything. But even siblings share anywhere between 50 to 100, okay? 100 being identical twins, right? And then it could be anything. So for transplant, people would have to take immunosuppressant, right? To suppress the immune system so they don't make antibodies to attack the antigens. All right, so let's go back to this. Type A blood, the genotype, right? We talk about it could be IAIA or IA small I. For B, it's IBIB or IB small I. So in other words, if I tell you I'm type B blood, you do not know if I'm homozygous or heterozygous. Okay, you don't know if I'm IBIB, the same, homozygous, or IB small I, heterozygous. Same thing for A. Okay? But if I tell you my blood type is O, then you know for sure I am little I. Same thing for AB, then I know the person is IAIB. Okay, let's take a look at the antigen or antigens on the RBC, red blood cell. There is my red cell with a biconcave disc in the center. If this is a type A person, then they would have the A protein on the surface, right? So what kind of antibodies will the person have in the bloodstream? That's right. They would have anti-B antibodies. Remember, you make antibodies against things that you do not have. Okay, there is no such thing as anti-O antibody, right? Because O is nothing, right? O is the absence of A and B. You cannot make an antibody that attacks nothing. That's why there's no anti-O. All right, we talk about who can we donate to later. Okay, what about here? If the person is B, then naturally they have B proteins on the surface of the red cells. And this time around, they will make anti-A, right? Anti-A antibodies. And if you are little I, what's going to be on the surface of the red cells? None. That's right. So no A, no B. Okay. Now it doesn't mean it has absolutely nothing. It could have other blood groups, right? It could have like RH on it, for example, right? It could have like other stuff. Okay. Just not A and just not B. And because they don't have A, they don't have B, we're gonna put they're gonna have anti-A and anti-B, right? Anti-A, anti-B.
Professor,
Yeah.
what about AB positive? I mean, they are universal acceptors.
Yeah, we're gonna talk about that. Okay, hold that thought. You might have heard of people say universal acceptor or universal donor, right? Like O negative, people say they're universal donor, right? But I'm gonna tell you why that is, not a very good name in my opinion. Hold that thought, okay, Ahmed. And if I forget to mention it, just ask me again. Okay, so what if IAIB, right? IAIB, then you're gonna have A and you're gonna have B, right? What kind of antibodies are we gonna make here for this person? Yeah, none, right? Okay, nothing. Okay, because otherwise you would end up attacking yourself, right? And we definitely don't want that. The only time, like anytime your immune system attack yourself, you would have what we call like an autoimmune disease, right? Okay, like lupus, for example, right? ALS, all these, or multiple sclerosis, all these are autoimmune disease. We don't want that. Okay, so who can we donate to? Let me go through the logic behind it with you first, and then I can tell you what the shortcut is. Okay, so first of all, when we talk about blood transfusion, you have to understand, I think I mentioned this in the lecture already, but let me reiterate. You have to understand we do not transfuse the whole blood. Okay, you know how in the movies you see, someone had stick a needle into their arm and then transfuse directly to another person. That doesn't work. Okay, like even if they both have the same blood type, it's not necessarily going to work, because if you do that, you're transferring all your white cells and stuff like that as well, and it could end up doing more harm than good. Okay, and so what happens is when you do transfusion, you have to separate the blood into plasma, okay, which is the yellow stuff, right? And then you have to separate it into red cells. Okay, and the rules for transfusing red cell and transfusing plasma, they are completely different. Okay, so to make it simple and not confusing, we are focusing on the transfusion of red cells alone. Okay, so when we say blood transfusion, we are talking about the red cells alone. Okay, not the whole blood and not the plasma, just the red cells. Are we together on that point? Okay, so what do we have to pay attention to when we talk about red cell transfusion? Okay. When we talk about red cell transfusion, we have to ask the following questions. Okay, what kind of antigens are on the donors? Donor is the person who's giving the blood. Okay, donors RBC. All right, you want to know what's on the cell's surface. Number two, you want to know what kind of antibodies, if any, is in the recipient's RBC. Okay. You want to know what kind of antibodies they have in the plasma. Sorry. I got distracted with the incoming email. Antibodies in the recipient plasma. Okay. And number three, the third thing you want to ask is, will the antibodies attack the incoming RBC? Okay, that's the logic that should be going through your head when you're trying to determine whether a transfusion is safe or not. So let's just do an example. Okay, let's just say we have A giving blood to AB. Okay, do you think this is safe or not safe? Let's just do a vote. Okay. Okay, I'm just gonna give you like 15 seconds for this. Okay, maybe 30 seconds. Go. If you think it's safe, go for A. If you think it's not safe, B. Safe. B. Not safe. Okay. You're gonna go ahead and do this transfusion or not? Okay, that was 30 seconds. Most of you answer is safe. Let's see why it is safe. Okay, so first, this means A is given to AB. So antigen on the donor's RBC. Well, if you're type A, you would have A, right? Okay, what kind of antibodies do you have in AB? Well, if you look at the chart, there are no antibodies, right? No antibody. And so when you do the transfusion, that's okay, because there will be no antibodies to attack the incoming cells. Therefore, it's a safe transfusion. Okay? Now let's take a look. So this is safe. Let's try another one. What if we go, A, B to B? Is A, B to B safe? Well, let's think about that. So on the surface of the incoming RBC, you will have A and B. And what kind of antibodies do we have for the recipient? If the recipient is type B, they would have anti-A, right? So anti-A antibodies will tag anything that has an A on it. So if you go ahead with this transfusion, the antibody will attack the incoming cells and it will result in hemolysis, right? We talk about it in class. And therefore, this is not safe. Okay? So in general, that's the thought process you should have if you want to think it through. But if you don't want to think and you just want to be able to get the answer right away on a quiz because time is tight and whatnot, you can use this transfusion chart to help you. Okay, so AB is on the top. Yes, first one is safe, Hema. Yeah, this one is safe, okay? Because the AB recipient has no antibodies, right? That's why they could receive anything, okay? They could receive from A, they could receive from B, and they could receive from O, okay? So A can give to AB, O can give to AB, and B can give to AB. And of course, AB can give to AB as well, right? Okay, and O can give to A, O can give to AB, and O can give to B. And that's why sometimes O is called universal donor. But again, I'm going to come back to that point later. Do you guys understand how to use this chart? Okay, the arrow tells you who you can donate to. Any question at this point before we, venture further? No? All right. Now, that's just ABO. Okay. Now, we also have to consider RH, right? RH blood group. So, long story short, negative can be given to positive, but not the other way around. Okay. And the brief explanation is this, right? If a person is positive, that means it has RH proteins on it, right? And therefore they will not have anti-RH antibody. And let me say that one more time, right? RH positive person will not have anti-RH antibodies, right? So that's why they can receive from negative, they can receive from positive, but not the other way around. If it's the other way around, the negative person, well, if you finish watching all the recordings, then you will know negative people will get sensitized when they receive positive blood, right? That means they will start to make antibodies, and that will be that would not be good. Okay, so only negative people can give to positive. Of course, positive can give to positive as well, okay, but not the other way around. All right. All right, let's see. A positive. If you're A positive, who can you receive blood from? Okay, let's focus on the A portion. Okay, if you are A, then you can receive from other A's, and you can also receive from O, right? Okay, O can give to A, right? And if the person is positive, that means they have RH, then they can receive from both positive people and negative people. Okay. Who can A positive donate to? So just focus on the A for now. Who can A donate to? Look at the chart here, right? A can donate to other A's, and A can also donate to AB, right? And the person is positive. Positive people can only donate to other positive people. Okay. Now this one is the same. I'm just gonna write it down. And if you don't understand it, you can ask me later on, or you can ask me now. Okay. But I want to know the AB positive. AB positive. Who can we receive? Who can AB positive receive from? Can anybody tell me? Okay, yeah. So a bunch of stuff, right? AB positive can receive from A, B, O, A, B. And all positives and all negative, right? Does that make sense? Seriously, like if you don't understand this, ask now, okay? It's confusing for some people, and that's okay, right? That's why we're having this tutorial, right? Okay, now looking at this, you're like, it's pretty sweet to be A positive, right? You can receive blood from everybody. And again, I'm talking about RBC, right? So I'm AB positive, right? Okay, but does that mean I'm universal acceptor? I can receive any blood transfusion? The answer is no, right? Remember, there are like 40 different blood groups, right? Okay, so just because I am compatible within all these blood groups, I have to check to see if I'm compatible for the other ones as well. So for example, we didn't talk about this in class, but I just want to say it as an example. There is another blood group called Cal. Okay, so some people are Cal positive, some people are Cal negative. So, if I am Cal negative, then I wouldn't be able to receive blood from people who are Cal positive, right? And so, that puts limitation in terms of who I can receive blood from. So when we say AB positive is a universal recipient, or when we say O negative is a universal donor, we are only talking about within the ABO and the RH blood group. Okay, I hope that makes sense. And unless it's an emergency, like if I don't give blood to the person right now, person's gonna die. Then people usually will match all of the blood groups. Okay, you send a blood sample to the blood bank, in the lab in the hospital, and they will match all the blood groups and make sure that you get a compatible blood. Okay, but if it's like an emergency transfusion, they don't have time to do the matching, right? We call it cross matching. If they don't have time to do all the cross matching, they will only match the ABO and the RH blood group. Does that make sense? Right. So back to your comment, right, Ahmed? I hope that answers your question. All right. Who can we donate RBC to if you're AB positive? Right, just AB positive, right? Okay. So I will post the rest of the answer, but is there any particular one that anybody want me to talk about right now? Otherwise, you can just check it online later. O, you want to talk about O? O positive or O negative? Donna? O positive, sure, no problem. So if you're O positive, who can you receive from? Well, if you're O, you can only receive from O. Okay. Right. No other arrow points to O, right? So you can only receive from other O's. And because you're positive, you can receive from O positive or O negative. Okay. But you can donate to a bunch of people. You can donate to O positive, A positive, B positive, and AB positive, right? Okay. Because positive can only give to positives, but O can give to all the other. Okay. Victoria wants AB negative. Sure. If you're AB negative, who can you receive from? Well, first of all, if you're AB, you can receive from all the blood groups, right? But you're negative. That limits. You only can receive from other negative, right? Who can you give to? If you're AB, you can only give to other AB. But since you're negative, you can give to both negative people as well as positive people. Okay. User, some another student, B negative. Okay. So B negative, you can receive from other Bs and O's, right? If you look at the chart here, B can receive from other B. You can always receive from your own blood group, and you can receive from O as well. And since you're negative, you can only receive from other negative. You can donate to other Bs or AB. Negative can donate to both positive people as well as negative people. Does that answer your question? Anything else? O negative, sure. We might as well do all of them. A negative here, I'm just gonna put in, it's the same case, right? A positive, AB positive, A negative, AB negative. If O has no antibodies, why can't they get A, B? So no. O has antibodies, right? Donna, O has anti-A and anti-B, right? That's why they cannot receive from any of the other groups, because their antibodies will attack all of these cells, right? If you give A, yeah, that's okay. I know it's confusing, the antibody and antigen, so no worries. O negative, right? For Emma. So who can you receive from? Well, if you're O negative, you're kind of out of luck. You can only receive from other O negative. Okay. And then you can donate to everyone, basically, within the ABO and the RH blood group. So A positive, negative, B positive, B negative, AB positive, AB negative. Because your cells has nothing on it, right? There's no A, no B, there's no RH. So there would be nothing to react against. And you can basically give it to anybody. All right. In terms of receiving, yes, you can only receive from negative. But when you donate, you can give it to the positives as well. Because negative people can give to positive, right? Because again, there's nothing on your cells, so nothing to react against. Okay? No problem. Now, just want to mention one thing. Okay, this I will not test you on it, but I just want to mention it. Right? It seems like it's like nature is not fair, right? If you're AB positive, you're so good, you can receive from so many people, and yet you give so little. And then for O negative, it's the other way around, right? It's the selfless one, right? You can give to so many people, but you can only receive from O negative, right? But remember, we are only talking about the donation rule for red blood cell. Turns out the donation rule for plasma is completely opposite. Okay, so when it comes to plasma donation and receiving plasma, AB positive would be the one that can only receive plasma from AB positive. Okay, and O negative turns out to be the universal recipient when it comes to plasma. Okay, so you know it's actually quite fair. Anyways, moving on. Why don't you guys take a moment and try to fill this chart in yourself, and we'll take it up in maybe like 2-3 minutes. Right? The donors on the left side here, and can you give A positive to A positive? Of course, it's safe because it's the same blood type, but you want to check A positive to AB negative and so on and so forth. All right. So take a moment, try that out. Okay, you know what? I'm gonna try to put you guys into like breakout groups, and see if you guys want to talk about it amongst yourself. All right. Let me see if I can do that. I forgot how to do that. Okay, breakout rooms. So try to talk about it yourself. Okay, with 24 people, let's just break it down into 6 groups. So 4 people per group. We'll come back in a bit. Okay.
Professor.
Hi.
Hi, him speaking.
Oh, hi. What's up?
In my group, no one's want to talk.
No, that's okay. Okay. I'll close the group soon. Okay. Thank
Okay.
you.
Okay, I will do it by myself.
Okay. Yeah, that's okay. We'll take it up soon. Okay. Thank you, though.
Okay.
Okay, are we all back here? Yeah, so I can actually see which group we're talking. There's a little icon that shows me who's talking in what group. So thank you for those who participated. Some of you might be a little bit shy to talk, and that's okay as well. Okay. In any case, does anybody want to share their answers? Okay. For the first one. Okay, here. Which one is safe?
Yes, it's all. I think it's all safe.
All
for-
of the here is safe, safe, safe.
Yes.
Yeah, okay. Let's take a look at it, okay? Over here, A positive is given to AB negative. All right, let's see if that's safe or not, okay? A positive given to AB negative. So can A give to AB? If you look at our transfusion chart, right? Okay, I'm just gonna redraw it here. Okay. A can give to AB. That's okay. That's safe. However, we must also look at the RH, right? Positive cannot give to negative. So therefore, this is not safe. Okay, even though A can give to AB, but because the RH is not compatible, then overall is not compatible. Okay, you cannot just have part of it compatible and ignore the rest. Everything has to be compatible for it to be safe. Does that make sense?
Yes.
Okay, great. Now let's try the next one here, right? A positive to B negative. Same story, right? A cannot give to B, and that fails already. And positive cannot give to negative, right? So that's not safe. And the last one is not safe either. Okay. So actually, that's the answer. I don't know how that compares to yours. But if you want me to elaborate and explain a particular one, please let me know. We can do that.
Hello, Professor.
Hi, Mohammed.
Hello, I just joined the class. Can you please elaborate?
You just joined the class. Can I please what? Sorry.
No, I just joined the class.
Uh-huh.
So can you please explain in the number 8 why is not safe? Why is not is it safe?
Okay, sure. I can choose one other example, okay? And you can watch the recordings for the earlier session for the explanation as well. But let's just do another one, okay? Let's just say B positive to AB negative, okay? So this one, B positive to AB negative, right? So according to the transfusion chart right here, right, B can transfuse to AB, right? Okay, that's what the arrow tells you. The arrow tells you who you can give to. So B to AB is okay. However, positive cannot be given to negative, right? It will cause them to start making antibodies, right? So for that reason, B positive to AB negative is not safe. Okay. Let's try O negative to, for example, O negative to A positive. Let's just do that one. Okay. O can be given to A. You can follow the arrow and you can see that is safe to do so. And negative can be given to positive, right? Because it has no RH on it. Therefore, there will be nothing to react against. Therefore, this is a safe transfusion. Okay, hope that sort of makes sense. Anything else? Does anybody want me to clarify any of these before we move on? Okay. A couple of questions here that I want to go through with you. Just a quick polls here. Go ahead for the first one right here. All right, these are quick ones, so I'll keep it short. Yeah, most of you got it right. It is the basophil that's responsible for inflammatory response. Okay, next one, number 2. Go ahead. Okay, again, I'll keep it at half a minute. Okay, this one is a little bit all over the place, but if you look at the activation pathway that we drew earlier, right? Fibrinogen is right here, and in order to activate that, we need thrombin. Okay, so thrombin is the correct answer. Okay, that's E right here. Okay, now this one is challenging. And make sure you understand it. You probably will see it on the quiz. I will relaunch the poll. Okay, I'm just going to give you another 30 seconds or so to figure it out. Okay, a lot of you chose 2. Let's see if that's correct. The MM blood group, which is like a new blood group that we have not talked about, but what's important is that it has 2 alleles on chromosome 4, designated LM and LN. Okay, if the heterozygous condition is codominant, how many different phenotypes will there be? Okay, so a lot of things to unpack here. First of all, what does it mean by heterozygous condition? We learned about this last semester, okay? But I just want, well, I mean, I didn't teach it to you, but then you should have learned it last semester. Can someone tell me what heterozygous mean? Yeah, different, right? Okay, so in the context of this, we have 2 alleles, LM and LN. So let's write down all the possible genotypes. Okay, what's the possible genotypes here? Just help me out here. Anyone?
A,
I'm sorry?
IA.
But these are the alleles though, right? Yeah, I understand you're
Yes.
trying to give me the ABO, right? But this is a different blood group. So
So double M.
yeah,
L-
so like it'll be LM, right? Double M, yeah. What's another one?
LM
Mm-hmm.
small n.
LN, yeah. And
Yeah.
then the last one is LN, right?
Yeah.
Okay, do we all agree on this? Do you all understand how I came up with these genotypes? I am just combining the alleles in various combinations. Okay, that's all the different ways there are, right? Okay. So let's talk about the phenotype, right? Phenotype is the actual blood type, right? So if it's LM, what kind of blood type would this be? M,
M.
right? Yeah, because there's only one possible outcome, right? There's only one allele, so it's just one blood type. And then this is also quite straightforward, is N, right? Okay. Now the tricky one here is this middle one, the heterozygous condition, the one that has different allele. Okay. Now the question says the heterozygous condition is codominant, right? What that means is both of them will show up. So not M alone, not N alone. The phenotype would be MN, pretty much like AB blood type, right? So for that reason, the answer is C. There are 3 possible phenotypic expression. So whoever chose C, there's one person who chose it. Good job. Bonus mark. But for everyone else, have I convinced you that this is the answer? Does that make any sense? Okay, I surely hope so. All right. Now, if the question says, if I cross this out, right? Okay, if I change the question, instead of saying if the heterozygous is a codominant, if I cross this out and I say, if I say if LM is dominant over LN, right? Then how many expression are we gonna have? How many phenotypes are we gonna have? Let's relaunch that. Okay, same choices. I changed the question now, right? I said if LM is dominant over LN. Now, how many different phenotype? You guys learned about these dominant recessive stuff last semester, right? Like the second half of the course. Just wanna, verify that. Guys get me a little bit worried here. Okay, thanks, Tina. All right. Anyone else wanna log in their answer before I take it up? All right. Does the genotype change? Does the genotype change? No. There's still only 2 allele, so it's still going to be these 3 possible genotypes. Okay, LMLM will still be M, NN will still be N. LMLN, however, because it says M is dominant over N, that means the M will cover up the N, right? So if it's LMLN, what's the new phenotype here? What is it going to be? Yes, it's M, right? Because the M is covering up the N. So how many different blood types do we end up having? Just 2. It's either M or N. Okay. Now I want you to think about that. Okay? And if it still doesn't really make sense, email me, talk to me, we'll sort it out. Okay. Let's try another question here. The transfusion, and then we're going to take a break. Okay. Select all correct answers. And try not to look at the chart that we fill in, right? Like just try to do it from memory, right? All right, let's end the poll. A negative can receive RBC from. If you are negative, you can only receive from other negatives, right? So that's why B and E will automatically not be the answer. And if you're A, you can receive from other A's, and you can receive from other O's. Just look at transfusion chart, okay? And you cannot receive from AB, right? Because AB would have a B on it, and if you are type A, you would have anti-B antibody in your plasma, and it will reject the blood. Okay. All right, let's take a 5-minute break, and we will resume at 1:43-ish, 1:43:45, 43:44. I'll see you in a bit. Hello, it's me again. Let's keep going. Number 9. Josh's father has type O blood. If Josh has type A blood and he marries Kat with type AB blood, find the expected blood type for their children. Okay, so we're going to want to draw a simple pedigree here. A pedigree is like a family tree, right? So we have Josh. Square is for male. So there's Josh. And his father is type O blood. Okay, so this is the father, Josh's father, and he's type O. Josh has type A blood, and he marries Kat. Okay, circle is a female. Kat is AB. And then we're gonna want to find out, the blood type of their kids. So over here, the father is type O, then we automatically know that he is little I, right? What about Josh? Josh is type A, so he can be IA or IA small I. So which is it? Well, if you look at his father is small I, right? So he has to pass on one of the little I, right? Which means Josh must have a little I. This little I came from the father, right? And remember, one allele is from each parent, right? So the IA is it's from his mom, and the small I will be from his dad. Okay. What about Kat? Well, Kat is IAIB for sure, because that's the genotype for AB blood. Okay. So if Josh is IA little I, then when he makes the sperm during meiosis, right, the chromosome separates, and half of his sperm would carry the IA allele, while the other half will be carrying the small I. And for Kat, half of her eggs will carry the IA allele, the other half would be IB. So when you go ahead and do the Punnett square, when this sperm fertilizes this egg, then the kid would be IA. These two combined would give you IA small I, IB small I. Right? So there will be a 50% chance of type A blood, right? Type A. 25% chance of type AB, and another 25% chance of type B blood. Does anyone have any question about this? Okay. Let's try the next one here. Now, this one is a lot harder. Okay. It asks you to list all possible blood type for question mark here. Has anybody tried this question yet?
Me.
Yeah, sure. Go ahead. Do you want me to, do you want to share the answer?
AB.
AB, yeah. Okay, so AB. Anyone else? Anyone else? Tina, what did you get? You got A? Okay. Anyone else? Okay. Thank you, first of all. I don't remember what the answers are. Okay, we're gonna do it right now, and we're gonna find out. Okay, Donna also says A. Okay, let's see. Now, to approach a question like this, you will first start with the blood type that you are certain about the genotype. There are only two blood types that you can be certain of the genotype, right? O and AB. Right, so over here, we got type O here. We're just gonna write down little I. Type A is going to be IA something. Type B is IB something, right? It could be IA or IA little I, right? Over here it could be IB or IB little I. We don't know yet, and we'll figure it out. Over here, it is going to be IB something, and over here is going to be IA something. So you write down what you can. Okay, for people who know that you know the full genotype for, you write down, and for the rest, maybe you can write down half of what you know. Then you gotta do some analysis. You gotta think about this and see who's passing on what gene, who's receiving what gene from whom. Over here, type O, we have a little I. Well, one of them must came from the father. The other one must have came from the mother, right? So that's how we know the grandparents, right? In this situation, they are both heterozygous. They could not have been IA or IB, right? Otherwise, their daughter is not going to be able to have type O blood. Is that okay? Just stop me anytime, all right? Let's take a look at this situation here. Now, where do we know for certain where this IA came from? Did the IA get passed down by the father, or did it get passed down by the mother, or we really don't know in this case? What do you think? Do we know for certain where this IA came from? Or not really sure. It's hard to tell. Not really, yeah. Okay, anyone else? Do we know for a fact that IA has to come from the mom or has to come from the dad? Yeah, it has to come from the mom, right? It cannot have came from the dad, because if it came from the dad would have been like AB, right? Dad is B. It's impossible for him to have an IA. So for that reason, it must have came from the mother, right? Okay, and we can trace it back even one generation further, right? This IA came from like grandfather there, right? Okay. And what about the other half here? Do we know what this other half is? Right, this other half right here. Well, all we know is that this other half came from the dad, but we don't know what the other half of the dad is. I mean, the dad could have been IB, could have been IB small I. There's no way for us to find out, right? And so this remains a mystery. We will not find out with the given information. Okay, we also will not know this for sure. Okay, so back to, but that doesn't matter, right? Because we're looking for the question mark here, right? So let's think about the other thing over here. So we know the IA came from her father, which means the other part must have came from her mother, right? Okay, so her mother can either pass on IB or little I, but we cannot know which one it is, at least not with the current information, right? So it could be IB or it could be small I. If she inherited the IB, then she would have been type AB blood. If she inherited the small I, then she would have been type A blood, right? So both of these are possible blood type for this person, right? With the given information that we have, both of these are possible blood types. Is that okay? Now I have another question for you. Hopefully it won't make this more confusing. I know it's confusing the way it is already. But I want to know, let's say they have another kid, right? Let's say they have a boy this time. Okay, they have a son this time. Okay. What kind of blood type does the son have to have in order for us to rule out one of these blood types? Okay. In other words, what kind of blood type will the son need to have in order for us to know for certain what kind of blood type this person has? Let's call this, like, I don't know, Mary or something. Okay, what kind of, yeah, okay, great, yeah, exactly, right? If they have a son and it turns out to be type O blood, then we know for sure Mary here is going to be IA little I, right? Okay. Other blood types is still a mystery. Okay, you still cannot rule it out with other blood type. Okay, but with an O, they would be able to figure it out. Okay. Now rest assured, it's not gonna get more difficult than this. You know, good luck, I guess. And if you still need help, as always, you know where to find me. Okay, we have some baby that's being mixed up here at the hospital. First of all, we do not use blood typing to figure out which baby belongs to which parent, right? That doesn't work. It only works in some cases. The most definitive way of figuring out which baby belongs to which parents, we do DNA fingerprinting, right? That is like 99.9% accurate. Okay, blood typing it's just a theoretical exercise. Okay, I think I might have done this one already, but you know, what the heck, let's just do it again. Now, in order for us to do these questions, you have to remember 4 things. Okay, 4 things to remember. I feel like I've also done this in the lecture. I don't remember. I made a recording like at the beginning of the pandemic and I haven't watched it. So if I'm repeating myself, I do apologize for that. We need questions to practice on. Yes, we will. Okay, I have some poll questions coming up. Okay, don't you worry. And it won't be more difficult than what we've done. All right. I promise you that. Okay, I'm not crazy, contrary to what some of you might think. So 4 things to remember. Number 1, okay, AB an AB person cannot have, what kind of kids can they not have? Okay. Doesn't matter who they marry. Okay. Oh, exactly. They cannot have O kids. Okay. That's because in order to have a type O kid, you must pass on one little I, right? And your partner will need to pass on another little I, right? Okay. So biologically, an AB person cannot have a type O kid, okay? Because you don't have a little I to pass on. Okay. Similarly, a type O person cannot have, what kind of kids can a type O person not have? Well, if you're type O, you can only pass on little I, right? So it's not possible to have an AB kid. Okay, biologically, I'm talking about, okay, you can adopt an AB kid if you really want one, all right? So O person can only pass on little I, and to make an AB child, you need to pass on an IA or an IB. Okay, Another rule, two O people, I guess. I don't know what the right grammar for this is. Two O people can only have, what kind of kids can they have? If you have two people that are type O. Yeah, you can only have type O kids, okay? The whole family is going to be type O, okay? They can donate blood to each other and everybody will be happy, right? Because type O is little I, and then when you pass it on, it's 100% going to be little I, right? Okay, same thing. Two Rh negative people can only have Rh negative kids. All right, these are the things you have to memorize. Remember, when you do these type of questions, okay? Do not start doing like Punnett square and stuff like that. Number one, that's gonna take like way too much of your time on a test, and we don't want that. And number two, it's actually more confusing when you do a Punnett square. Okay, so let's take a look at this situation here. We want to start with people who have type O blood or type AB blood. Okay, so couple number two is a good couple to start with. Okay, first of all, they are both type O, so automatically, you know, anybody that's not O will not be their baby, right? Remember, type O can only have type O kids. It says rule number 3, and they are both negative, so that means they can only have negative babies. So that's not their baby, and so baby L is theirs. Do you see the logic there? Right? You don't have to do Punnett square. You just think about the rules and then you do process of elimination. Okay? So that's gone. This baby has been claimed. Next. Okay. The mother is AB. If the mother is AB, the mother cannot have an O baby, right? Type O baby is not possible. Okay, so that's gone. The father is type O, and according to rule number 2, he will not be able to have an AB baby. So that baby is not theirs. And L is already taken. The only one left is baby H. Okay. Next one has an O. Only 2 babies left to take home. And we know type O cannot have AB baby. So that must be I. And then the last one.
Professor,
Yep.
for number 3 couple,
Yep.
so there should be answer is H.
Yes.
So the rules number 2 and which one will be applicable? 2 and what?
2 and 1, right? Because
Yeah.
O cannot have AB and AB cannot have O, right?
Okay.
Yeah. So that's the answer. All right. Now, this goes without saying, but I'm gonna say it anyway. You should try this out on your own again, right? Like later on after we hang up. Let's try number 12 right here. Okay, number 12. So again, we want to start with parents that have AB type blood, type AB blood or type O blood, right? So first one, AB, which means we will cross out the O, right? Type O, which means we will cross out AB. Okay. So is it baby G or baby H? What do you think, huh? For couple number 1. Anybody want to explain? Baby H, yeah. Okay, can someone explain to me why?
Because both are negative.
Exactly, right? Both of them are negative. They cannot have positive. So that has to be baby H. Okay, so baby H is taken. Next one, A positive and O positive. So O means they cannot have AB, right? So that's gone. And then we have O positive and A positive. So based on the information we have right now, we don't know. We don't know which one it is. Okay, it could be F and it could be G. We can't decide right now, but we know for sure it's not E. I'm just gonna put F and G here for now. Okay. Couple number 3, type O. The mother is type O, so not E. And then we have 2 babies left, O positive and A positive. but neither the mother or the father has an A allele, right? None of them has an A allele. So it's impossible for them to have a type A baby. So F is theirs, okay? Which means F cannot be couple number 2, and it's only G. And so the last one that's left is E. Okay, that's it. I hope that makes sense. And again, right, only certain combinations would work for these kind of matching, right? You will not believe how much time it took me to come up with these questions. I have to make sure that, you know, they actually worked out. But in real life, right, if there is any confusion about which baby belongs to whom, they do, you know, DNA fingerprinting. Okay, that's that. More practice, you were asking. Let's do that. Yes, let's do this one first. Which of the following is possible? Okay. Seems like it's singular here. I am going to launch the poll. Okay, go ahead. So just choose one answer. Yeah, I think. Excuse me, it's only one. Oh yeah, these should be A, B, C, D. Okay, that's okay. I'm sure you'll figure it out. Oops. Yeah, I think most of you got it right. Okay, here O negative and AB negative. So they are both negative. So I cross out all the positives. Okay, O cannot have AB. So that's the only answer. Right? You see how quickly you can do these questions if you memorize the 4 rules, or you just cross out rather than looking for what the answer is. You just cross out all the ones that is not true. Okay, and then hopefully you're left with one choice. Okay. All right, now this is what you've been waiting for, right? Okay, this is the pedigree. What is the blood genotype of John? Here's John. Okay, here's John. I'm gonna let you try that. Okay, relaunch the poll. I'm gonna give you 2 minutes for this. Okay. Let's take this up. Oh, that's okay. I wouldn't know which one you choose as long as you have an answer in your mind. Then that's okay. All right, let's see what happens here. So we're trying to figure out what John's blood type is, his genotype specifically. So let's start with the people that we know. So Sue is little I. Mary is IAIB. Ed here is little I. And everything else is just, we only know half of it. All right. So Sue is the child of Harriet and John, which means one of the small I came from Harriet and the other small I must have came from John. All right. Let's take a look at Mary. Mary is also one of the children, and IA came from Harriet. And where does IB came from? Well, IB has to be from John. Right? IB has to be from John. Cannot be the other way around because Harriet doesn't have a B to pass on, right? So the A has to be from Harriet and B has to be from John. And so the correct blood type for John is, what is it? IB small I for sure. Okay. Let's try one more time. What is that?
What
Yeah.
about Muna?
What
Muna.
about who? Mona?
Muna?
Yeah, that's the next question. Why don't we
Yeah.
why don't we figure that out? Okay, go ahead. Okay. Let's take this up. It's the same tree as before. I'm just gonna go back here and use the same information. So Harriet is the daughter of Ed and Mona, and the small I came from Ed, right? Because Ed doesn't have an IA, and therefore the IA came from Mona. Okay. But do we know what the other half is? Well, we really don't know. Okay, so it could be IAIA, it could be IAIB, it could be IA small I, right? So in other words, it could be anything. We really don't know. So the correct answer would be need more information. Okay, over here.
Professor,
Yeah.
why we don't know? Because John have IB and I.
Yeah, but John is not their son, right? John is the son-in-law, right?
Oh.
Yeah, it's okay. It's
Okay.
I
Sorry.
thought it's the son, John.
John
So,
is not there. Yeah.
yeah,
So
I
yeah.
thought-
Yeah. Okay, but it's
Sorry.
only their son if there's a direct line coming from it, right?
Yes,
Yeah,
exactly. Yeah.
Yeah. No problem. No problem. Okay, so it's need more information. Okay. We all good now? Okay, we got to move on. We have a lot more stuff to cover. All right, next one here is the blood typing, right? Okay. So basically you have three wells on the plate, and the wells contain antibodies in it, and you're going to add the blood sample to the wells, and you want to see if there's a reaction. Okay, so there is a play like this. Okay, we actually would do this in a lab if we are in person, and then there will be like a well, right? So each of these well has antibodies in it, and you would add the blood to it and see if it clumps up or not. Okay, so when you see this little flaky stuff here, we call this agglutination. Okay, agglutination, which is just like a fancy word for clumping, right? So clumping that occurs as a result of cross reaction. Okay, so cross reaction is when the antibody tags the antigen. To the RBC. They cause them to clump together and then they break apart, right? Hemolysis, right? So why would the antibody clump to the RBC? Well, they would clump together if the a blood group matches the antibody, right? So the fact that the sample reacts with antibody A tells you the sample contains type A blood. No clumping with the antibody B, no reaction with antibody B tells you the sample does not have the B antigen. Reaction with the RH tells you it has RH, therefore it's positive. So basically, whenever you see a reaction, that's the blood type that you have within the sample. So no reaction with anti-A, that means no A. Reaction with anti-B and anti-RH, this person is B positive. No reaction with anti-A, no reaction with anti-B. So that means the sample does not have A and it does not have B, which means the person is type O. O positive though, because of reaction with RH. Reaction with anti-A and anti-B, that gives you AB. No reaction with RH, that's AB negative. I understand these are a little bit harder to see. And for those of you who have the workbook, you might not have these pictures in it. But I promise you the one on the quiz will be a lot clearer. So here, no reaction with A or RH, only reactions with B. If I zoom in a little bit, you can see, you know, the little clumping here. That means it's B negative. Reaction with all of them, that's AB positive. AB negative for this one, and O positive for that one. Does anyone want me to explain further? I will post the answer for the fill in the blanks and you can check it on your own. Probably it's more efficient that way. I do want to move on to long answer questions because it's, you know, some of these could be confusing. Does anyone want me to take any of them up? Any special requests? Number 7, sure. Anything else?
We're gonna do application questions, right?
Yeah, we will.
Yeah.
Anything else besides the application 6? Okay. Okay, I might as well do 5 as well. And again, I explained this already in the video, but it's a confusing concept. Let's just quickly talk about it again. All of them are related anyways. Okay, so let me try. All right, this is just going to be a condensed version. Okay, for the full version, just go watch the video yourself. All right. First of all, you have to understand this. When I say a person is type A blood, then tell me again, what kind of antibodies do they have in the plasma? If someone is type A blood, what kind of antibody do they have in their plasma? And type B, exactly, right? Okay, now for our purpose, we can think of those people who are type A blood. They are born with the anti-B. But as you will learn next week in the immune system, that's actually not always the case when it comes to antibodies. Let me give you an example, right? Your body does not start to make anti-chickenpox antibodies until after you have been exposed to chickenpox, right? That makes sense? Right? Similarly, right? We do not have antibodies against COVID unless we either A, caught COVID, or B, got vaccinated, right? So a lot of the antibodies, even though we have the potential, we have the capability of making it, we actually don't start making it until we are exposed to that particular disease or to a particular antigen. Am I making sense here? So for ABO blood group, you are pretty much born with those antibodies. All right. However, when it comes to RH, you will not start to make anti-RH antibodies until you have been exposed to RH positive cells. Just like the chickenpox that we talk about, right? You don't make antibodies until you are exposed to chickenpox. All right. So on that point, let's quickly write that down. RH negative individuals cannot make, well, we'll say will only make anti-RH antibodies after being exposed to RH positive cells. Okay. This is called, what is this called? Help me out here, please. What is this called? When a negative person has been exposed to positive cells. What do we call that?
Sensitization.
That's right. We call this sensitization. Now, I want to emphasize that sensitization is not restricted to RH group. Okay, all the other blood groups that we haven't talked about, all of them, a person can get sensitized to it. Okay, as long as you are negative, you can potentially get sensitized to the positive version of that blood. Remember earlier I talked about like Cal, right? So if you're Cal negative, you can become sensitized to Cal positive blood. Right? If you're MN negative, you can get sensitized to positive blood. Okay, so each of the blood groups can potentially have a sensitization problem, but we are focusing on the RH one. Now what the next question is, over here, right? Exposed to positive cells. How on earth does a negative person get exposed to positive cells? How? Okay, we talk about 2 ways in the lecture. Any of you remember what it is? How does a negative person get sensitized? Yes. Okay, number 1 is pregnancy. Okay. Can someone explain more further? What about pregnancy? Every time a negative person gets pregnant, they become sensitized, or what? What kind of babies do they have to be carrying in order for this to happen? Yeah, so this would be an RH negative mother carrying an RH positive baby, fetus, right? And there is mixing of blood. Okay. So the mother is Rh negative, maybe the husband is Rh positive, and that causes the baby to be positive, carrying an Rh positive baby. And the baby's blood got into the mother's bloodstream, right? That would cause sensitization. All right, what's another way to get sensitized? Okay. What's another way to get sensitized? That's right, transfusion. Okay. Rh positive blood is transfused into an Rh positive, Rh negative person. Okay, maybe it was done as a mistake. Maybe they had no choice, right? The person is dying, bleeding out. We only have positive blood. We know they had not been previously sensitized. Go ahead with the transfusion, and you know, it would save them one time, right? Remember, if you are a negative person, you get one free exposure, right? The first time you're exposed to positive blood, it's okay. You won't die, right? You will only become sensitized. You will have the antibodies. But once you have the antibodies, you cannot be exposed again, right? Okay, because then you will react. So does everybody understand sensitization and how you get sensitized? Okay, so both female and males can get sensitized. But sometimes people get so hung up on the pregnancy idea, they think men cannot get sensitized, but men can get sensitized through transfusion, right? So can women, right? So can anyone, basically, right? If you're negative and you get transfused with positive blood, you are going to be sensitized. Okay, so how does this relate to hemolytic disease of the newborn, HDN, right? Okay, let me just quickly explain again, right? We have the mother's blood here. This is the mother's blood. Mother's blood, and let's say our mother is Rh negative here. Okay, so we're going to draw some red cells. Okay, and this is the first baby. This is the baby's blood, the fetus blood. Okay, we'll just say baby's blood. And this is Rh positive. Okay, because the father maybe is Rh positive, so you know they have a positive baby, and the separating the mother's blood and the baby's blood is of course the placenta. The placenta. Placenta is the yes, it separates the mother's blood and the baby's blood, but it also connects the mother and the baby, right? That's, you know, how nutrients go from the mother and the baby, right? And for those of you who don't know, the placenta actually gets pushed out after the baby gets pushed out, right? Again, usually, it's thrown away, right? Okay. Just on a side note, I just remember a story that I read last year, and it was kind of funny. I think it happened in Toronto. The police, they found placenta, like human placenta, in the park, and they were really concerned because they thought, you know, oh my goodness, someone must have given birth to a baby in the park on their own, and left, and they thought, you know, the baby must have been abandoned or something, you know, and so they did the search, and then, you know, and then they made it onto the news, right? News report we're talking about it, and it was on the newspaper and all, and then a couple of days later, the owner, I guess, or the owner of the placenta, the person came out and said, you know, it was her placenta, but it was from, like, you know, many years ago when she gave birth at home. And so at that point, she gave birth, and then, you know, the placenta came out, and she didn't know what to do with it. So she decided to just put it in her freezer. And so after, like, you know, so many years, she was clearing out her freezer, and then she found the placenta, and she was like, oh, rather than throwing it away, maybe she'll just leave it at the park so that the coyotes can eat it or something. That's exactly what she said, right? And then there was just a big confusion. Anyway, there was no baby harm in the process, but it was quite a funny story. In any case, back to the serious stuff. The placenta separates the mother's blood and the baby's blood, and in our situation here, right, the baby is positive, right? So I'm gonna draw her her blood right here, and we're gonna put some RH on it. Okay, this is an RH positive baby, right? And so what happens is sometimes the placenta can get damaged, like during pregnancy if there's some kind, if the mother falls or something, and if not, during delivery, during the actual labor delivery process, it's possible for the placenta to get damaged, and in that case, the baby's blood will go into the mother's bloodstream. Okay, so now you have the baby's positive cell in the baby's bloodstream, in the mother's bloodstream. Okay, and that caused the mother to become sensitized. Okay, and what that means is her immune system is going to get triggered, and she will start to make anti-RH antibodies. But that's not going to happen like immediately. It takes about 2 weeks, 1 week or 2 weeks, in order for the antibodies to be produced, right? Think about like last time you got sick, right? Before you start feeling better, you're going to be feeling lousy for 4 or 5 days or even a week, right? That's the time when it takes your antibodies to be produced and fight off the disease, right? So it's not going to be an immediate process, which means by the time the mother produces all the antibodies, right? These are anti-RH antibodies. By the time the mother produces the antibodies, the babies would have been delivered, okay? And then it would not affect the first baby. And the antibodies will clear all these RH cells, and that will be it for the first pregnancy. Are we still together on this first pregnancy? Okay, no trouble for the baby. By the time the antibodies are made, baby's already out. Okay. Now, second pregnancy. Okay, let's say the mother has another child and it's positive again. From her previous pregnancy, she was sensitized, and so she already has all these antibodies in her, right? All these anti-RH antibodies. And what happens is now these antibodies can actually cross the placenta, and they will go into the baby's bloodstream, and they will attack the baby's blood. Okay, so that will result in massive hemolysis in the baby. Okay, all the baby, well, a lot of the baby's red cells are gonna burst. Okay, and to deal with all these red cells dying, the spleen and the liver has to work extra hard, right? So if you look at the PowerPoint slides, hemolytic disease of the newborn is characterized by enlarged spleen and enlarged liver, right? There's going to be edema, swelling, right? There will be jaundice, right? Because you probably remember from lecture, when red cells breaks down, they release the hemoglobin, and the hemoglobin gets broken down into bilirubin, right? Which is this yellowish stuff. So you have a lot of hemolysis going on, then there will be a lot of bilirubin that builds up. So the baby will be very jaundiced when they are born. And so collectively, right, that condition is what we call hemolytic disease of the newborn. Now, sometimes it could be very severe. In the case of RH mismatch, it could be quite severe, and they might have to do transfusion for the baby. Sometimes it's kind of mild. Okay, even the ABO blood groups can have a little bit of mismatch. Let's say the mother is type O and the baby is type B, right? Then the mother has anti-A and anti-B, right? Okay, so my wife is actually type O, and my daughters, both of them are type B. And what happens is her anti-B can cross the placenta and start attacking the cells. And so both of my daughters were born quite jaundiced, right? And then, but it's rather relatively mild, right? Certainly not to the same degree as the RH mismatch. So they just had to put them in the UV light, the Billy light, right? The chamber with the purple lights, and that helps remove the jaundice. But that's basically what hemolytic disease is, and how it relates to sensitization. Okay. Now, how do you prevent this? To prevent this, we are going to use RhoGAM. Okay, RHOGAM. RhoGAM is artificial antibodies that attack RH cells. Okay, so what that does? Okay, let me just clear up all these drawing. I might as well erase the whole thing and start again. Okay, so baby is RH positive again. There is your placenta. And then there is the mother's bloodstream. Okay, so let's go back to the first pregnancy. Right? First pregnancy, the RH positive cells went into the mother because placenta was damaged either during pregnancy or during delivery. Okay, but what happens is the mother can receive a RhoGAM shot sometime during her pregnancy. Okay, 28 weeks in pregnancy, and they could receive it again immediately after delivery. Okay, and so what the RhoGAM does is, first of all, it's an antibody that attacks Rh, and if the mother already has the RhoGAM in her system, these are the RhoGAMs. Okay, the RhoGAM will immediately destroy all the positive cells that are making their way into the mother bloodstream. So the RhoGAM will remove the Rh positive cells within the mother before her immune system is activated. Okay, so the purpose of RhoGAM is to prevent sensitization. Okay, it does not reverse sensitization. It does not cure sensitization. It prevents it from happening in the first place. Does that make sense? Now you should have you should be wondering, you should have a question about RhoGAM. Okay, you might be thinking, well, how come the RhoGAM does not go into the baby bloodstream and attack the baby cells, right? So remember, these are synthetic antibodies. The people who make it makes it really big. They make the antibodies so big that it will not be able to cross the placenta. Okay, it will just remain in the mother's bloodstream, and it will not affect the baby at all. Does the mom show signs? How do they know to give it to her? Yeah, so when a person gets pregnant, usually the mother would do a blood test, and the father would also do a blood test. If the mother is negative and the father is positive, they will recommend the RhoGAM. Okay. Now, I hear what you're saying. If the mother is negative, right? Okay, there's only one way to be negative. But the father is, if the father is positive, let's say the father is positive, right? Then in this case, 100% guarantee their baby will be positive, right? Okay, then the RhoGAM is definitely necessary. But if the father is positive, negative, then it's only 50% chance that the baby is positive, right? Okay, but there is no easy way to tell whether the father is positive or sorry, let me rephrase that. There's no easy way to tell whether the father is homozygous or heterozygous. Okay, there's no easy way to check whether the baby is positive or negative. Okay, so they will usually just recommend receiving the RhoGAM shot anyways. Okay, even if there's just a 50% chance. The only time where you definitely do not need the RhoGAM is if, number one, if the mother is positive, then this is out of question. Okay, you do not give RhoGAM to positive people, because then the antibody will start to attack the positive cells, right? And that's not good. You would not give RhoGAM to people who are negative, and then the husband is also negative, because again, right, it's impossible for them to have a positive baby, right? My husband was never tested while I was pregnant, so it was not a standard test. Yeah, I mean, it's supposed to be, you know, standard, but then you know how healthcare is, right? Sometimes it's inequitable access, right? So for some people, it wasn't even brought up. But you know, it's usually a safe thing to do. But maybe they know that the mother is positive, and therefore there's no need to test for the husband, right? Okay, it's only when the mother is negative, then they have to find out what the husband is as well, right? Does that make sense? Yeah. Okay, great. So that is that. Okay, that's everything. Let's go back to our application question here. Bone marrow transplantation can be used to treat leukemia. Speculate the effects of bone marrow transplant on the person's blood type. Justify your answer. So you know, the bone marrow is where all the blood cells came from, right? Remember the big word, right? Yeah, sorry, go ahead, Mavis. Oh, okay. The hematopoiesis, right? Okay, that's the process of creating new blood cells, and all your blood cells came from the bone marrow. Okay, and leukemia is basically uncontrolled division of white cells. And so by replacing the damaged bone marrow, the mutated bone marrow, with healthy bone marrow, you can stop the leukemia. Okay, so when it comes to bone marrow transplantation, the blood type has to be compatible. So let's say the person is AB, and you do the bone marrow transplant, you can give bone marrow from a type B person. Okay, but afterwards, what happens is the blood type would change from AB to B because all the new stem cells from the donor, they would only produce the type B blood, right? Okay, so in that respect, it's possible for the blood type to change after bone marrow transplantation. All right. I actually had a student 6-7 years ago, I think. She told me she had leukemia when she was younger, and after receiving the transplantation, her blood type actually did change. So it's a common thing. Next one. Sally's first child was born with hemolytic disease of the newborn, HDN. She learned about RhoGAM from her friend. Sally is now pregnant with her second child, and she is requesting to receive RhoGAM shot from her doctor. Is her request valid? What do you guys think? Should
No.
she? No, why not?
Because she already sensitized.
That's right. She already sensitized, right? Having the RhoGAM will not reverse that. Okay, the RhoGAM only prevents your immune system from being triggered, but once it's triggered, the antibodies are already there. It will not prevent it from happening. Okay, so what can she do in that situation? Well, nothing much they could do really. They just have to monitor the situation, and if it's really serious, sometimes they can do what we call like in utero transfusion. That means they could do blood transfusion while the baby is still within the mother. That's of course not without risk, but sometimes that's the only thing they could do. All right. So that is that. It's a lot of stuff going on in this lecture. Certainly take your time to review the material. The quiz is next week, right? Next Tuesday, I believe. Yeah. It'll be available from 6 a.m. all the way to midnight. I will post the information for you after class. When I post the recording and everything, I'll post the information for the quiz for you as well. If you need an accommodation, if you are registered with CALS and you need accommodation for extra time, please send me the documentation and I will factor in the extra time for you. Otherwise, I'll see you next week for lecture 3. Okay, thanks again. Have a good one.
Professor,
Yeah, go ahead.
okay, I already booked an appointment for ISP. Last semester I had that, but this semester I already booked an appointment. The appointment is 19th, and I already got the documentation from my doctor. So what can I do now for this next quiz? I tried to get the appointment early, but they are busy. They are busy. I tried that appointment like almost 3-4 weeks before.
Yeah, okay, talk to them and see if they can issue you like a temporary statement, and then tell them to maybe like contact me. You have my email, right? And then maybe they could show me something from your last semester that you were on file. That way I can go ahead with the to process your
No, you have my information.
Oh, I did. You email me last time?
Yeah, last time I did that.
Okay, can you send me what you sent me last semester again, just so that I have it? Is that okay?
Yeah, okay, I will email you.